Semua ni 50% dari 400 lebih saya lagi.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera and a very good morning. Distinguished guests, ladies and gentlemen, we are about to begin. May I have your attention, please? Our program will start in a few minutes. I would first like to remind all of you to keep your microphone at mute while the program is running so as to minimize sound distraction for everyone's benefit. Thank you. Good morning, ladies and gentlemen. On behalf of the organizing committee of the webinar, From Metagenomics to Environmental Spectrum, I would like to extend a very warm welcome to all of you. My name is Nora Shirin Muhammad Jamil. Together with Miss Andrea Amanda, we will be your MC for today's event. It is our privilege to have you here with us today. We are now ready to begin the webinar. We hope you will find the program we have lined up for you to be fruitful and engaging.
Ladies and gentlemen, we will start our program with an opening prayer. Let us pray for a blessing from God Almighty from this gathering today according to our respective religions. Praying started. Praying finished.
To continue with this morning's program, we will proceed with Malaysia's and Indonesia's national anthem. Now, it is my honor to invite Dean of Fakultas Biology, Universitas Kristen Satyawacana, Dr. Luciawati Dewi, to deliver her opening speech.
Okay, good morning, all the participants. First of all, I would like to give my thanks, especially for UE TM, Faculty Science Kegunaan, and this is wonderful collaboration. So we could have the event together. I sincerely wish that we could know each other better through all this event, so that we could have our long-lasting partnership together with
Ibu, you are muted.
I'm sorry, yeah, stay on mute, yeah. Okay, good morning, ladies and gentlemen. First of all, I would like to give my thanks, especially for Faculty Science Kegunaan, UITM, for this wonderful collaboration. And so we could have this event together. I sincerely wish that we could know each other better through this event, so we could have our long-lasting partnership together between Fakultas Biologi UKSW and also Fakulty Science Kegunaan UITM. Also, we would like to express my gratitude to Professor Farida as a Dean of Fakulty Science Kegunaan and her colleagues that are willing to make this collaboration for both university. Thank you for also the speaker who has been willing to give a material presentation. And also this morning, let this event mark beginning of our great partnership so that we could hold many events together, such as a joint seminar. And also maybe next time we can join research together and also student exchange and also as a guest lecturer. I expect that this partnership also continues for years to come and that it brings mutual benefit of both our sides. So to know each other better, I would like to share some information about Fakultas Biologi UKSW that it's established in 1962 and currently has 3 program study. And also I would like to introduce my structural office committee and also another information from my set. So I would like to share my PPT presentation about 3 minutes maybe, yeah. Yeah. Wait a moment. Yeah, this is my official committee. This is I'm a dean, and also we has a vice dean or head of study program for biology. That is Dr. Andes Susahyo MSc, and also we have head of biology department, Dr. Hewan Chuhan, MAM PhD, and we have head of study program for the education of biology, Dr. Marisa Christina Tapilo, SSI MT. This is my vision. We focus in bioscience, especially in applied science, in food, industry, and environmental sector. And also we have a biology education. So in the undergraduate, we have 2 program study. That is biology education and biology for as a pure science. And our mission, we have 5 mission. This is the important things that we have a focus research in agriculture, food, environmental, microbiology, and also management of environment, climate change, biodiversity, biomaterial, and phytopharmaceutical. Local-based learning media, this is in the education of biology. And the focus of community service, we have a community-based ecotourism development, and also we have a Tempe innovation training. This is my special focus in Tempe research. And also we have a biology learning training based on local wisdom. This is our network, and I put in UITM in here also. And I want to focus in this slide. Maybe next time we can have a collaboration, especially in student exchange, collaboration in research, and collaboration in lecture or practical work. This is, I hope that in UITM, Professor Farida also agreed with my suggestions like this, and applied science in my site is about fermented food, ecoenzyme, ecotourism, and also learning media. This is, we have undergraduate students and focus in this area. And also we have graduate students. In graduate student, we have focus in molecular biology and also in the environmental management and phytopharmaceutical. We have a program about the double degree. Additional around 36 credit or SKS, we call SKS, to get education and science bachelor degree and also science and education degree. So we, the student we can give to to degree, yeah, in biology and also in education biology. And also
Muted, Ibu. You accidentally mute.
yeah, we have undergraduate in 144 credits. We also have acceleration program. This is 5-year program, and the student can also follow from undergraduate to graduate level. So if the student study for 5 years, we can give MSc degree. Yeah, this is our site. Professor Farida, maybe we can next time can speak each other discussion and which one we can collaboration, and then we will make a understanding MOU together. Thank you very much for this morning, and be healthy all the participants. Thank you.
Thank you, Dr. Luciawati Dewi, for the introduction. I'm pretty sure everyone is really excited, and I hope this will initiate a collaboration between FSG and Fakultas Biology.
Without further ado, it is my pleasure to invite the Dean of the Faculty of Applied Science, University Teknologi Mara, Syah Alam Selangor, Professor Dr. Hajah Farida Surana binti MD Yusuf, to deliver her opening speech. Dear Prof. Farida, the screen is yours.
Thank you, Andrea. Thank you so much, Ibu Lucy, for inviting us to be part of the collaboration. And we are really looking forward, yeah, for this student exchange, staff exchange, and also to the MOU, so that we can plan of what you have presented just now. Yeah, like, okay, now about faculty science gunaan, it's a slightly different, because you have faculty of biology. Faculty science gunaan biology is a program in this faculty. So at the moment, I'm the dean. Okay, so since it was first established in 1967, the same age as mine, and when UITM, it was previously known as ITM, Institute of Technology Mara. When ITM got its university title in 2000, so now it is called Faculty Science Gunaan. Our faculty is huge, one of the most biggest faculty in UITM, and we have our faculty's branch campus at almost all the states, yeah, throughout the country. So now we have 10 branch campuses of Faculty Science Gunaan. It's huge. Okay, so this is our strength at the moment. In Shah Alam only, we have 215 academic staff, 23 admin staff, and 54 lab staff, of which we have 102 labs altogether. Okay, so this is the number of students at the moment, yeah, throughout from pre-diploma to PhD. And we have here in FSG Shah Alam now, as the main campus, we have bachelor's degree, of which we have 6,697 students. Yeah, 76 of them are lady, yeah, and only 24% men. Yeah. Okay, so what we have here in FSG for year 2021, we have 2 pre-diploma courses, 8 diploma courses, 20 bachelor honors courses, 1 PhD, 14 Master of Science by Research, 2 Masters of Science by Coursework, and 3 Masters of Science mixed mode. Yeah, this mixed mode only for 1.5 years. For Master of Science, 2 years. Yeah, for Master of Science coursework, also 1.5 years. Okay, so our pre-diploma programs, We have a pre-diploma program for science, and science now year by year 2018. Now we have STEM C. Yeah, we add arts track in that particular science program. So we introduce one new pre-diploma program, consists of 2 semesters. What is so special about UITM, Ibu Lucy and all the participants, we have 60% of our students come from B40 group of population, where it means that the income of the family is less than 4,000 ringgit Malaysia. So what we have for this kind of students that come from that particular family, they will be given free tuition fees, free accommodation, and monthly allowances. Yeah, so that's why we have huge number of students that we want to groom, to give knowledge to them. Okay. Okay, for diploma programs, we have polymer technology, microbiology, industrial chemistry, food technology, wood technology, textile and clothing technology, science, and industrial hygiene and safety technology. And Bachelor of Science honors that we have here in the main campus, Shah Alam, we have School of Biology. So we are now here with you, Ibu, and the rest. School of Biology, we have Biology program, Biomolecular Science program, Marine Technology program, Applied Microbiology program, Biodiversity Management. So we also have School of Chemistry and Environment, School of Physics and Materials, School of Industrial Technology, and we also have here, Ibu, double degree, where we have Textile Science and Fashion Technology Design. Our collaboration with art and design, arts and science. So we have double degree. On this double degree, we are the first in UITM to come out with this double degree. Okay, for postgraduate program, we have Master of Science coursework, Science and Food Technology, Environmental Technology, and we also have Master of Science mixed mode, Textiles, Applied Microbiology, and Applied Biology. And the rest what we have is Master of Science and Doctor of Philosophy. Okay, so for these programs in FSG, we have accreditation of our academic programs from Textile Institute, Royal Society of Chemistry, and MBOT. Most of our program, 5 our diploma program, and 8 of our degree program accredited by MBOT, Malaysian Board of Technology. Okay, so we send 2 subjects, for subject ranking from Faculty Science Gunaan, and we first joined in 2017 where we have chemistry, 1 subject to be ranked, and then good results. We got 2 subjects ranked in 2019, which are chemistry and material sciences. And we are planning to send 5 subjects from Faculty Science Gunaan. Maybe this one also we can co-join with Fakultas Biology, UKSW, Ibu. So we have material science, environmental sciences, physics and astronomy, and biological science for 2025. Okay, so our strength in the faculty is research. So we are one of the research faculty. So we have 2 journals from the faculty. We have our editorial board from this faculty, which are Science Letters and Junior Science Communication. We are welcome, our counterpart to publish in these particular journals. Okay, and these are our collaboration initiative, MOU and MOA thus far. So these are all the companies. And these are all the universities. So we're looking forward to have UKSW, one of our list very soon. Okay, so the innovation and commercialization by our faculty, I'm very proud to announce that our researchers come out with products, and these products are now everywhere in the Shopee, in a mall. Okay, when I explain for this one, Dr. Aziza, Dr. Siti Roha, and Dr. Fadila, they have come up with sauce nanas, jenama paina. Prof. Madya, Dr. Ramah Muhammad with the polymer product, the degradable bottles and plastics, and Dr. Muhammad Azri, the product with minyak wangi, perfume, Dr. Azri's perfume. And Prof. Dr. Rosie, penghasilan songket, ya, we have teknik jacket teknik. Dr. Aida Firdaus bersama produk kopi, Una Coffee, ya, then this is very good coffee, low in caffeine but high in protein. And last but not least, Dr. Khairul Aswa from Algae Cream Cheese Wafer with partner companies. Okay. Okay, that's all from Faculty Sains Kegunaan for this time. Thank you so much.
Okay, thank you, Prof. Farida.
Ladies and gentlemen, we will now proceed with the video profiles of each faculty.
We wake up by the sunrise and you're staring back at me. I hold on to the moment as we cast it out to sea. And the lights come on, reflecting off your skin. So burning stars are all we are, ignited from within. It's your heart and soul, it's your flesh and bones. Don't you let it go. You're the storm I find, bursting through my sky, taking all control. It's your heart and soul. It's your heart and soul. I will pull you from the places that you've seen. Fly away from the darkness to a paradise we dreamed. And the lights still shine, reflecting off our skin. So burning stars are all we are, igniting from within. It's your heart and soul, it's your flesh and bones. Don't you let it go. You're the storm I find, bursting through my sky, taking all control. It's your heart and soul. It's your heart and soul. It's your heart and soul. shining, we'll be shining, we'll be shining tonight.
Selamat datang di kota Salatiga, kota kecil yang sejuk dan nyaman untuk belajar. Kampus Universitas Kristen Satya Wacana Salatiga menjadi salah satu tujuan untuk kita menuntut ilmu. Kampus yang hijau, sejuk, dan nyaman untuk belajar, serta berbagai keunggulan yang dimiliki. Salah satu keunggulan itu berada di Fakultas Biologi UKSW yang mempunyai slogan Biology for a Better Life.
Fakultas Biologi didirikan pada tahun 1962, yang dimana kami memiliki staf-staf pengajar lulusan S2 maupun S3 dari dalam dan luar negeri dengan slogan Biology for a Better Life, di mana kami selalu ingin melakukan kreativitas dan inovasi di dalam basis-basis yang ada di dalam ilmu biologi. Dengan 3 program studi, yaitu program studi S1 Biologi, kedua adalah program studi Pendidikan Biologi, dan yang ketiga kami juga ada Magister Biologi S2 Biologi. Untuk menyongsong Merdeka Belajar, kami juga telah bekerja sama dengan mitra-mitra kami yang terkait di dalam ilmu-ilmu biologi.
Di Fakultas Biologi UKSW, kami sebagai mahasiswa diajarkan untuk berpikir kritis, analitis, inovatif untuk menuju lulusan yang ahli di bidang biologi atau menjadi tenaga pendidik yang kreatif. Di Fakultas Biologi UKSW juga, kami diberikan informasi serta pendalaman ilmu pengetahuan yang efisien serta efektif melalui teori-teori di ruang kelas atau melalui pengamatan yang lebih mendalam seperti praktikum dan observasi langsung di lapangan, seperti magang dan sebagainya.
Kuliah di Fakultas Biologi UKSW itu sangat menyenangkan karena lingkungan kampusnya yang sangat tenang dan nyaman untuk belajar. Terdapat fasilitas kelas yang sangat memadai, serta sarana laboratorium yang sangat mendukung dalam bidang ilmu biologi.
Tenaga pendidik yang berkompeten dan berkualitas juga disediakan di Fakultas Biologi UKSW, sehingga tak jarang kami dapat mampu mengeksplor lebih dalam terkait dengan ilmu biologi. melakukan riset dan penelitian bersama para dosen, sehingga kami semakin ahli dan profesional di bidang biologi.
Perpustakaan kampus UKSW menyediakan pada kami banyak buku tentang biologi. Serta terdapat juga buku dan jurnal yang lengkap terdapat di perpustakaan UKSW.
Kami memiliki beberapa lab yang menunjang untuk kegiatan praktikum dan kegiatan penelitian. Antara lain adalah lab biologi dasar, lab mikrobiologi, lab biologi molekular, kemudian ada lab aquakultur, dan lab pengolahan limbah baik cair maupun padat. Dan yang terakhir adalah lab BSL level 3, yaitu Biosafety Lab, yang digunakan untuk kegiatan riset di dalam bidang biologi molekular. Kami juga sudah ada di dalam peminatan bioentrepreneur, di mana dengan basis ilmu biologi, maka mahasiswa lulusan bisa melakukan kewirausahaan yang kreatif dengan basis-basis ilmu biologi yang ada. Biology for a Better Life akan meningkatkan taraf hidup kita pada lulusan-lulusan Fakultas Biologi ke depan.
Ladies and gentlemen, we are privileged to have with us today two inspiring keynote speakers. Our first keynote speaker is from the Faculty of Applied Science, UITM. It is my honor to invite our moderator, Dr. Marisa Christina Tapilau, to introduce to us Dr. Muhammad Shafiq Azmi. Dr. Marisa, the screen is yours.
Thank you for having me as a moderator in this session. I would like to invite Dr. Muhammad Shafiq bin Azmi. Yes. Dr. Muhammad Shafiq bin Azmi is a senior at School of Biology, Faculty of Applied Science, University Teknologi Mara, Shah Alam. I would like to introduce his academic qualification. He started the academic qualification with Bachelor of Science from Medical Lab Technology, UTM. After that, he studied about at Master of Science at Health Science, UTM, and also after that, he graduated a PhD as a PhD at his competence is Metagenomics. I would like to review about he has many research and publication. I would like to mention two of his research and publication. The first one is about uncovering probiotic-mediated gut microbiota modulation in hypertensive mice. And the second one is influenza disease burden and cost estimates in Malaysia. He has many publication. Maybe I would like to mention three of his publication. The first one is about at a journal in Singapore Medical Journal, collaboration with many researchers. The title of his publication is about the effect of proton pump inhibitor on the human gut microbial profile in multi-ethnic groups in Singapore. And the second one is about the effects of single lactobacillus casei strain, Sirota, and probiotic cocktail. The third one is about the technology advancement enabling the link of gut microbiota with disorder. Right now, Dr. Muhammad Syafiq bin Asmi will present his presentation about a metagenomics approach to unravel the association of gut microbiome with health and disease. So, Dr. Muhammad Syafiq bin Asmi, please, time is yours. Thank you.
Good morning and Assalamualaikum everyone. Thank you, Dr. Marisa Christina, for the very kind introduction of me. And first of all, I would like to thank Fakultas Biology, UKSW Salatiga, and also School of Biology, Faculty of Applied Sciences, UiTM, for this opportunity and also the invitation. Begitu senang to share some knowledge with all of you, rakan-rakan di dua negara on the metagenomics. So today, my presentation will be on topics of metagenomics. So I think, should I share my slide now, or Mr. Azam will control the slide for me?
Please.
All right. Thank you, Mr. Azam. So for this particular session, allow me to share with you some knowledge. Right, I'm a junior lecturer here and just joined UiTM for 3 years. And my interest is basically on the topics of metagenomics, pretty much related with the health and disease. So today, the session is about metagenomic approach, okay, to unravel the association of the gut microbiome with health and disease. And for this particular session, next, Mr. Azam. For this particular session, there will be 4 content or outlines that I wish to share with all of you. First, of course, I would like to introduce a bit about the concepts of the metagenomics. And then I'm going to share with you common approaches and also workflow pipeline that have been used in metagenomic studies. And I wish to actually share as well with all of you the typical analysis pipeline and also how people interpret the data in the metagenomic studies. And if the time permits, I wish to share with you an example of the application of the gut microbiome into health condition, which is obesity and also maternal health. All right, next. Now, allow me to introduce to the metagenomic. Maybe the terms metagenomic will be jargon to some of you. So metagenomic can be defined as a study of a collective genome of a microorganism recovered directly from their natural environment. So their natural environment could be from the soil, right, from the sea, oceans, or even a specific anatomical sites of a human body, for example, skins or your oral microbiome or nose.
And this technique is actually allow you as a scientist to bypass, okay, the need of isolation and lab cultivation of individual species. And this technique is powerful, meaning that to provide information on the microbiodiversity and ecology of the specific environment. Next. And when we talk about the metagenomic, the main objective is actually to understand the dynamic nature of microbial population and their impact on the environment and human health. Because why? Because microbes play an enormous role in the processes that relate to generally every aspect of our planet. For example, harvesting energy and nutrient from our diet. And usually, when we design a study of metagenomic or in the metagenomic study or data, there will be three main scientific questions that need to be answered. Okay, as a scientist, we want to know who is that, meaning that what is the population or composition of the microorganism in that particular ecosystem that we have interest on to research. And then what are they doing based on the occurrences of the gene content or functional gene that been encoded by this particular microorganism in that particular ecosystem. And now the third will be how are they doing? Basically, it's more into functional analysis and all that. So all these three questions is the main scientific question or research hypothesis that drive our study design to validate or to explore the role of the gut microbiota in various metabolism, including health and also disease. And I'm going to share with you the typical study design for each of these scientific questions. Next. When we talk about the metagenic study, right, with the aim, as I mentioned, to determine the abundance and identity of the microbe in any particular ecosystem, okay, recovered directly. We can use two approaches, either 16S sequencing or shotgun metagenomics. For the 16S sequencing, actually, we rely on the amplification by PCR on the prokaryotic 16S ribosomal RNA, and then we sequence it using next-generation sequencing, for example, Illumina, MAS6, and all that. And the presence of this particular gene in all bacteria, and this gene is actually a universal conservation of some regions, so that it allows you to use it as a marker in reconstructing phylogenies, okay, due to the specificity of the some 16S gene variable region. And this allow you to make a bacterial identification at sufficient informative taxonomy rank, right? And whereas for the second technique, shotgun metagenomic is kind of perform whole genome sequencing instead of a single bacteria, but then you sequence all the entire microorganism in that particular ecosystem directly. So in order to do that, you have to shed or fragment the DNA into a smaller fragment, say 500 base pair or 200 base pair, before sequencing. It's pretty much depending on your technical requirements of your sequencer or sequencing platform, such as if MAS6, 500 base pair or 250. And this technique is allow you to comprehensively sample all gene, not only 16S, but then functional gene or gene content in all organisms in that particular ecosystem. Next. This figure or this table is shows you some advantages and also disadvantages between these two techniques. Okay, principally, as I mentioned, for 16S, we focus on the 16S as in target on the 16S gene, whereas for the shotgun is focused on whole genome-based phylogeny, whole genome phylotyping. Advantages for the 16S is actually provide a fast survey of a large community, because it's just a 16S, you just extract the microbial DNA from the sample, then you perform the library preparation by amplifying the 16S gene, and then you load in the sequencer. And it gives you a good for first round survey, whether that particular ecosystem has a specific microorganism or they have a different profile or population structures and so on and so forth. And it's quite cost effective. In Malaysia, I think the cost will be about 100 to 150 US dollars per sample. Whereas for the shotgun, no amplification bias because you don't perform a 16S amplification prior to the sequencing. And it provides a community-wide assessment because this shotgun metagenomic sequence all the genome content, and you can get the information on the bacterial composition and the complement functional gene presence. However, for Of course, these techniques has a disadvantages for the CCNS. It might have an application bias and sequencing error because it's rely on the PCR application sequencing base. Whereas for the shotgun, it's a computationally expensive because the massive data, one sample for the shotgun metagenomic will generate about 3 to 5 gigabase data. So the assembly of metagenome is quite challenging because due to the uneven coverage, because you cannot expect that, you know, in that particular ecosystem, you will get the so-called similar abundance of all the bacteria, okay, across these species. Next. This figure summarize again, how does one mine the microbiome? Again, it's depending on your approaches, whether CCNS or shotgun metagenomic. For the CCNS, you, as I mentioned, we have to amplify and sequence the CCNS. Then you have to perform bioinformatic analysis by data binning, meaning that based on the sequence that been generated, we need to group this sequence into a similar sequence as a group. We call it as a OTUs, Operational Taxonomy Unit. And each OTUs, for example, here, OTU 1, 2, 3, 4 will be BLAST against several databases, Green Gene, RDP, CIVA, and then CBI. And then the number of hits will represent as the relative abundance of each OTUs, you know, as to reflect the composition or diversities of that particular species or bacteria. And in normal situation, when you perform CCNS molecular characterizations on the single bacteria species, you only blast against NCBI. For the metagenomic, you need to blast against several databases to validate your similarity search. Whereas for the shotgun metagenomic, you sequence the community DNA and then you have to perform again data binning based on the CCNS RNA to identify the species. And then in the same time, you have to perform gene annotation, okay, to predict the functional contents of the gene based on the KEGG, SEEDS databases. And the data presentation for both techniques will usually start with the bar chart to represent, you know, the abundance of the species and also the occurrences of the gene. Right. Next. And remember the three main questions that I've mentioned to you just now. All right. For the first, who is that? Basically, to answer this particular question, usually reports in metagenomic studies will present a taxonomy and function overview, okay, by presenting the relative abundance of bacteria at each taxonomy hierarchy, for say from the kingdom to the genus or species, and the annotated functional category will be reflected in the percentage. On your right hand side, there are two pie charts. You know, the first pie chart tells you that from this data or from this particular ecosystem, 98.9% compositions are bacteria, whereas the second pie chart tells you that, you know, 34.1% of bacterial composition belongs to the alpha Photobacteria. Right. So the bacterial distribution usually will be presented in the bar chart or histogram. So each slice indicate the percentage of reads with predicted proteins and also ribosomal RNA genes annotated to the indicated taxonomy level or functional categories. Right. Next. Because gut microbiota, because of microbiota is actually corrective microorganism in that particular ecosystem, and they are very has high adaptability, okay, towards any changes in the environment. So they have trait of inherent plasticity. So with this inherent plasticity of the gut microbiota, easily modulated changes and all that, you need to perform comparative metagenomics analysis in order for you to actually identify the biologically important and to make an inferences out of it. All right. Usually, when you want to identify which bacterial genera or taxa that has the biologically important, you need to perform comparative metagenomics at 3 different scales, from alpha diversity, beta diversity, and also gamma diversity. Alpha diversity is actually described the species diversity within a community at a small scale or local scale within one ecosystem. Whereas for beta diversity, describe the species diversity between two community or ecosystem. For example, in figure 1, I'm sorry if the figure is not very clear to you, but you could see that there are 4 types of a cluster. You know, the green, blue, the baby blue, and also the red one. So it tells you that from figure 1, the gut microbiota community for the green one, meaning the obese profile is totally different from the red dotted profile, which represent lean obese profile. So this tell you that the beta diversity is different, okay, between the gut microbiota in obese and also lean profile. Right, next. And then second question will be, what are they doing? So for this particular questions, you know, our aim is to elucidate the functional capacity of the microbiome in the speech condition after you manage to profile the composition of them, and secondly, we want to catalog, okay, the collection of the genes in the microbiome, and then again to estimate the abundance of the specific metabolic pathway and any shift or changes after given a specific intervention modulations. All right, and we have to perform bioinformatics analysis, you know, by binning the rates oriented against databases, and then the number of reads that map to each gene is used as proxy for its abundance. For example, on your right hand, there will be a figure showing the gene profile of microbiome for Pygmy loris. Pygmy loris is the actually animal. So you could see here the abundance of occurrence of the gene correspond to the metabolism involving with the carbohydrate, amino acid, protein are the highest in this particular animal. Right, next. And to answer the third question, how are they doing? Basically, it's more into like functional study where required multi-omic approaches. You know, not only that, just metagenomic. Imagine just now you managed to profile the composition of the bacteria, and then you manage to annotate the functional gene of it, and you know that what is the types of the gene that has been encoded by this bacteria or this particular ecosystem, and putatively their functional role in that particular ecosystem and how they react to the environment. Now, to validate that, you have to utilize transcriptomic proteomics, metatranscriptomic analysis, you know, to distinguish the active from inactive members of the microbiome. In order to do that, you have to extract the RNA, total RNA, proteins and all that, you know, to validate whatever function that gene function that had been annotated previously. All right, next. Now, I would like to share with you one of the example application that been carried out in UiTM. The project is related with the obesity. Why obesity? Because Malaysia is one of the fattest country in Southeast Asia, and statistically speaking, recently, you know, 1 in 2 Malaysian adults are either obese or overweight. So when we talk about the obesity, it's a multifactorial disease, and we cannot say one single factor contributing to the progressions of the obese. And usually, when we talk about the obesity, it could be due to the genetic polymorphism, genetic traits, you know, because of the SNP, short nucleotide polymorphism, or due to the impairment of certain gene regulated in the satiety or lipids, such as gene regulation, or it could be due to the environment factors, you know, classical factors such as poor dieting habit, sedentary lifestyle predisposes towards obesity. And I think for the past 10 years, compelling evidences shows that the gut microbiota are different between obese and also lean, and few functional studies shows and prove that, you know, the role of the gut microbiota in the development of obesity. What is gut microbiota? Microbiota is actually collections of microorganisms residing in our gastrointestinal tracts. It comprises of bacteria, virus, parasite, and also fungi. And the density could be up to 10 power of 11 microorganisms. And the species for the bacteria itself could be up to 1,000, right? And since this gut microbiota is so diverse in the ecosystems and encoding millions of functional genes, functional gene, so they involve in nutrient and also energy harvesting in the host, and it helps our body to actually harvest the nutrient byproduct of this particular bacteria. And with the traits of inherent plasticity of the gut microbiota, which easily modulated based on our dietary pattern, so it provides a new avenue for us as a scientist to actually treat obesity, or through by application of biotherapeutic strategy using probiotic, prebiotic, or through food management, right? To combat obesity. The idea is actually to try to modulate the obese gut microbiota structure and hoping to ameliorate the obesity and related comorbidities. Right, next. So to prove this concept, we developed an obese animal model, right? G1 is obese control rats that been fed with high fat diet for 21 weeks. G2 is an obese rat that been treated with probiotic Lactobacillus casei, strain Shirota, or known as LB13 here. And then the G4 is the lean rats, or control lean groups. And after 12 weeks oral supplementation with these probiotics, we found that probiotic able to actually reduce the final body weight, energy intake, subcutaneous fat, total fat weight, and improve the total cholesterol levels, TG, and also leptins in the obese rats significantly as compared to the obese control. Now, this part and our interest to further profile the gut microbiota, whether the gut microbiota are different between these three groups. All right, now let's see. Next, Mr. Azam. All right, here in this figure, you could see that the PCAs of gut community structures. Okay, and here you can tell easily from your naked eyes that the gut microbiota composition in obese rat, as highlighted in the green colors, are different from the gut microbiota profile in the lean rat, as highlighted in red color, or red dotted on the PCA. And then after 12 weeks of probiotic supplementation, you could see that the profile of the gut microbiota in obese rat are totally different from the control. And it might give you some ideas that, all right, based on here, the profile is different. Most probably their genetic content or functional gene will be different. All right. Okay, next, Mr. Azam. Before to answer that, we observe, this Venn diagram shows there are 21 genera that have been affected significantly by high fat diet, whereas there are 35 bacterial genera that have been affected significantly by the probiotic supplementation. Among the 21 genera that have been affected by high fat diet, enrichment of 70 genera, as highlighted in red, are positively correlated with the obese phenotype, higher body weight, subcutaneous fat, and all that. Whereas a reduction of 2 genera, as highlighted in the blue color, negatively correlated with the obese phenotype. Whereas among the bacterial genera that have been associated with the effects of the probiotic LB13, we observe enrichment of 16 genera, highlighted in the brown color, positively correlated with the improved obese phenotype, right? Whereas reductions of 10 genera, as highlighted in purple color, negatively correlated with the improved obese phenotype. And among the 16 genera that have been found significantly increased post-probiotic supplementation, we found that enrichment of cholesterol-lowering bacteria such as Bacillus, Eubacterium, and also Rhamnococcus might provide natural hypocalcemic effects under LB13 treatment. That's why you could see that from the phenotypic observation, the total cholesterol, leptins, and body weight subcutaneous was significantly reduced as compared to the obese control. These changes in the composition of the gut meta suggest changes in their metabolic capacity in obesity. Okay, now, as our hypothesis saying that it may suggest changes in their metabolic capacity, how we validate this? All right, next. Next slide, please, Jazam. All right, here, true enough, from our gene annotation, we found that probiotic modulated and lean gut microbiome had lower occurrences of gene related to the fatty acid metabolism and nutrient transport, lipid and nutrient transport. And you can see that those boxes that are highlighted in gray, meaning significantly reduced in either probiotic treated with the probiotic L-betatin or the lean control as compared to the obese. All right. All right, next. And second application that I wish to share with you is on the maternal health. So this is more recent studies. So this studies is titled gut microbiota in pregnant Malaysian women. So we compare the profile between trimester, BMI, and so gestational diabetes status. So we recruited 38 Malay women from gut flora studies in UiTM Medical Center, Sungai Buloh. And then we collect their stool sample, and then we perform the C-CNS ribosomal gene amplification. We compare the gut microbiome profile between trimester 1 versus trimester 3, normal versus abnormal or higher BMI, and the third will be between healthy and also those who are having GDM, gestational diabetes. Right, next. And this figure 2b shows that the composition of the gut microbiota between trimester 1 and also trimester 3 are not different. So although you could see some trends, but it's not significantly different, right? So we can say that the composition of the profile is actually quite similar between trimester 1 to up to trimester 3, right? Next. And then when we compare and detailed inspection between patients that having having GDM versus healthy. So in the figure 4b, we found that the profile of the gut microbiota between patient having GDM as highlighted in the red dots and also green dots are totally different with the healthy one who having non-GDM status, blue dots and also cyan dots. So, and based on this, we can say that several bacteria compositions or several bacteria actually contributed to the significant observation on 4B, which are you see from 4C, which is VIP, very important projection score. So all these bacteria is actually found significantly high in GDM pregnant women, right? Okay, next. Then next, the third certification, and we found that based on the figure 6b, the compositions of the microbiome between mild underweight, which with red dots, and also the normal BMI with the green dots are totally significantly different as compared to the pre-obese highlighted in the cyan dot, and also the obese blue dots in the PSA. So we can say that the composition between obese and also normal BMI of pregnant women are totally different. And based on the VIP score plot in the figure 6C, there are several bacteria that have been found significant high in the underweight. And all these bacteria is actually give you an idea about the possibility of their roles in the developments of this particular health condition between pregnancy or even during obesity in animal model, right? To validate this function, maybe we have to isolate and also to test their capacity in the cholesterol conversions and all that. But here from this, you can see that lots of quite numbers of bacteria that be associated or be reported to be elevated in during inflammations or in diabetics cases of patients. So it gives you some hints that this particular bacteria might have a role in the inflammations, right? Okay, next. So in summary, based on this data, the metagenomic approach could determine the dynamic nature of microbiome population and their impact on our health, meaning that you can compare between healthy and unhealthy or between two conditions of health. Today, most studies are more descriptive and also correlation study, as I presented just now, rather than causation study. So we could get an idea of which bacteria may play a role in the observed phenotype. So like the case of obesity and also GDM mellitus, gestational diabetes knowledge on the composition of the gut microbiota provide an avenue for us to apply biotherapeutic, probiotics or natural product. For example, supplementation of high fiber diet could be suggested, okay, to serve as a nutrient to the gut microbiota, which could modulate the composition of the gut microbiota, and hopefully we could control the cases or the prevalence of obesity or reduce the health risk or health consequences among GDM of women. All right, next. So before I end my session, I would like to welcome all of you, right, to submit your manuscript to our homegrown journal, Science Letter. This Science Letter is actually indexed by National Citation Index, my site. Next. So our area of interest is as depicted here: material science, environmental science, chemical sciences, food science, and medical science. I'm one of the editorial board. I'm as a managing editor. Right, next. So please visit our website, and you may check the guidelines, and also you may submit your manuscript through the online system. So I welcome all of you to submit your manuscript for the next year publication. So, okay, next. With this, I would like to thank Ministry of Higher Education Malaysia to support my study before, and then UiTM, Faculty of Applied Sciences, Faculty of Pharmacy, I Promise UiTM, and also Faculty of Medicine UiTM. With this, thank you.
Okay, thank you, Dr. Shafiq, for your wonderful presentation. We will open the discussion. I see there are 2 questions in the chat column. The first one coming from Mr. Jacob Octosea. Maybe Mr. Jacob could open your video so Dr. Shafiq will know you. He's one of our staff in Fakultas Biologi. Mr. Jacob, maybe you can open your camera so we cannot know each other. The question of from Mr. Jacob is about gut microbiota types are related to the ethnic groups. So how to differentiate people among ethnic groups in mixed population like Malaysia or Indonesia?
All right, thank you, Dr. Jacob, for the question. Very interesting. Yes, of course, Indonesia and Malaysia, the social demographic is quite pretty similar. We are multi-ethnic countries, and obviously, when we talk about the microbiota, it's pretty much influenced by diets. And of course, when we talk about diet, there is a custom, budaya, tradisi makanan dan sebagainya. And I would say there are a few reports, okay, on this from. I think from the Indonesia, there is several published studies on the kids as well, and also for adults, and there is a significant difference between the population of the gut microbiota among the ethnic group. Yeah, and it's pretty much related with the diet as well, because as Asian, right, our diet, our meal pattern is actually very, very, very rich in terms of nutrients and everything. We can mix in one plate with carbs, protein, and another proteins with the chili and everything. So obviously I would say the gut microbiota population is very unique. But how to differentiate, meaning in order to prove these scientific theories, you need to recruit, different ethnics of subject, Chinese, Indian, or Malay, and you need to actually do food frequency recall, and then you have to record all their dietary habit and meal pattern for the past like 3 weeks, and from there you have to analyze, how many cups of proteins and all the macro and macronutrients. And then from there, you need to ensure that they are actually had a similar lifestyle sort of, you know, in terms of social demographic and all that, in order for you to really see the differences, you know. And obviously there will be much different between urban and also suburban pupils. And you need to be more particular about the study design if you wish to see the difference. But obviously there will be differences in the profile of the gut microbiota among multi-ethnic due to the diets and also the lifestyle, right? Okay. And then Dr. Marisa, can we move to the second question?
Okay, the second question coming from
Dr. Samsul.
Dr. Samsul, can the gut microbiota act as an immunomodulator in obesity metabolic disorder? For example, adipose tissue macrophage.
All right, thank you, Dr. Samsul, for the very interesting question. There are few studies that able to show that, you know, modulation of the gut microbiota reduce the inflammation level, interleukin 6, 7, and they relate this particular concept with the gut-brain axis, where, you know, relate with the inflammation in the particular in the host. And with regard of whether it can be used as immunomodulator in obesity metabolic disorder, theoretically, I would say yes, but we need to identify how to modulate the gut microbiota as an immunomodulator in obesity. It could be used a prebiotic or probiotic and see the changes, and from there, how this particular bacteria, as I presented here, ataupun able to reduce the certain inflammation marker. And then when you measure the subcutaneous adipose tissues and all that, how actually this particular bacteria influence, you know, this particular adipose tissue to release the macrophage that lead to the inflammation. And it require multi-omics platform, as I mentioned. Maybe need to support the finding with the gene expression, you know, on the adipose tissue itself, and then to quantify the macrophages involved and all the inflammation markers that need to be evaluated pre and post of treatments or modulation of the gut microbiota. And from there, maybe to validate further, you need to isolate the several bacteria species that be identified as significantly increase or reduce upon modulation. And then we can try again in the germ-free mice to really see that whether this particular phyla or bacteria is actually could modulate the inflammation, especially in controlling the adipose tissue macrophage. I hope that my explanation could give some ideas or like some knowledge a bit to this all of us.
Okay, very clear. Thank you, Dr. Shafiq. Very clear. Thank you.
Yeah.
Thank you, Dr. Shamsul.
Welcome. No problem.
For the participant, is there any question for Dr. Shamsul? If there's no question, I will like to I have one question for you.
Okay. Dr. Marisa.
Such an interesting presentation. I see about the emerging methods and how are they doing about the genomes. Could you explain to us more clearly about the method, the emerging method? I see in your presentation about the one side about the difference, the other side about the emerging method. Could you explain the difference of two of this method?
All right. Okay. Basically, when we talk about the advancement methods in the sequencing, for example, right? Here, metagenomics is one of the platform that been used or technology that been used. And just now, Dr. Marisa, based on the presentation, I mentioned that, you know, in order for us to validate the function or to answer the functional aspect of the microbiome, we need to employ multi-omic strategy like transcriptomic, you know, metaproteomics and all that. So if we learn about the center of dogma, where from the gene, translate to the proteins and actually they act accordingly, and then to involve the metabolism of that actually regulate the metabolism or certain physiological situation. So once now in the metagenomic, you already identified a certain composition of the bacteria, and then you know the functional content of that particular encoded in the bacteria. So now you need to know what sort of protein that have been secreted, and then what sort of messenger mRNA that has been expressed, which gene has been expressed. Because when you perform the functional gene annotation, you just annotate, whether it's an express or not, or whether it's a function or not, you cannot say much, because it's just putative based on the blast and annotation. So you can say that the gene is there, this microorganism encoding this particular gene. But whether it's a function expressed or not, you have to extract the microRNA from the for example, stools and all that. You have to do the transcriptomic study to validate whether this particular gene really expressed, and then from there you have to see what sort of a protein that has been will produce or has been produced, and then you have to perform the proteomic study as well. So basically you validate the data from one to another, and yes, baru you boleh kata this particular gut microbiota can actually modulate the obesity metabolic disorder, for example, or this particular gut microbiota with these changes and profile able to actually reduce or control the secretion of the macrophage from adipose tissue, for example. So it's require multi-omics to actually to wrap up your scientific inquiries as a whole. So that's what I mean by multi-omics and multi-studies. Yeah, I hope I answer to your inquiry, Dr. Marisa.
Certainly. If there is no question, I will like to have another question for you. I see the last study, the subject of your study is a pregnant woman, right?
Yes,
about probiotic. Is there any relation about the probiotic activity and also the hormonal activities? So you choose the pregnant woman. Is there any relation about it? Thank you.
When we talk about the hormone, is quite huge topics, right? That I agree with you. When we talk about the pregnancy and all that, the hormones it must be involved, and that's why actually our data just show it's just a glimpse part of it, whether this gut microbiota is a difference or not during pregnancy and with this kind of heart condition. And in the between, there is a involvement of the hormone. So in order for us to explore a bit more, we need to actually evaluate the hormonal status and need to validate again using the model, to really ensure that there is a relation between. But again, as I mentioned, for now, based on the medical records and everything, we can just do correlation analysis based on statistics and everything. Maybe there is a significant correlation, but in order for us to confirm it, and in order for us to say that this particular profile of gut microbiome is the causation for this particular hormonal imbalance, leads to you to have GDM or obesity during pregnancy, we need to do a questionnaire study. Again, back to the first question from you, Marisa, to validate. And yeah, this is one of the gap of knowledge that, recently lots of studies have been published about gut microbiome, but it's more on correlation. So we need to confirm it using functional study. And yeah, I really hope that, we could collaborate somewhere, and I hope more scientists could give an insight on this. Yeah.
Okay, thank you for your response about my question. It's only about something in my mind about after this see your presentation, such an interesting presentation. I think after this, I will like to say thank you very much, Dr. Shafiq Asmi, for your wonderful presentation and give us a wonderful insight about another topics about metagenomics. Yeah. For Dr. Shafiq, maybe we can collaborate after this. I see about your journal science letter. Maybe I will like to submit one.
Yeah, we
article.
welcome all of you to submit your manuscript to us.
Okay. For the next session, I would like to give this session to Ms. Norashiran Muhammad. Thank you.
Thank you, Dr. Marisa, and thank you, Dr. Shafiq, for the insightful presentation. Over to you, Andrea.
Ladies and gentlemen, moving right along, it is my greatest pleasure to invite our next moderator, Dr. Samsul Bahrin Gulam Ali, to introduce to us Dr. Hewan Johan, our second inspiring keynote speaker. Dear Dr. Samsul, the screen is yours.
All right, thank you very much, Ms. Andrea. So good morning, everyone. So now we move on to the second presenter of the session. So let me give a brief introduction about Dr. Johan. All right, how are you, Dr. Johan?
Okay, fine, thank you.
All right, good, okay. So Dr. Johan obtained his doctoral degree in environmental science from Griffith University, Australia in 2002. Then he was awarded William and Lily Foundation grant to pursue his postdoctoral in environmental risk assessment from the same university, which completed in 2017. And currently, he is an associate professor in Satya Wachana Christian University, UKSW. So his expertise involved mainly environmental toxicology, risk assessment, and management. His research publication mostly involved the toxicity and hazard assessment of chlorobenzenes, which are intermediates in producing pesticides, dyes, and other industrial chemicals. So without further ado, I invite Dr. Johan to deliver his talk with the title Aquatic Risk Assessment of Highly Lipophilic Toxicant. So the platform is yours, Dr. Johan.
Thank you.
Welcome.
Yeah, Regina, please show the slides. Yeah, morning or afternoon, everybody. Actually, my presentations, has some relationship with that presentation by Dr. Shafiq. Why Dr. Shafiq tries to help us reduce obesity, help to cut the numbers of obese person. My presentation, especially dealing on the other direction, try to get fatty fish as the indicators of environmental pollution. So we share some common ideas about lipids, both in the obese people and in the obese fish or the fatty fish, whatever. Yeah, sounds good. And my presentation today is aquatic risk assessment of highly lipophilic compounds. And my presentation is based on two parts. The first part is the results of my latest research in risk assessment, and the second one is a proposed framework, my framework or proposed idea for the next biomonitoring program in aquatic risk assessment. Next slide, please. And we see, this is on my paper, bioconcentration, toxicity, and hazards of chlorobenzenes in the aquatic environment. Normally, we starting with biology, ecology, and some chemical or chemistry analysis. Of course, for the biology people, we are more focused on the concentrations of these toxicants on biota, on fish, in fish. So that's why I started with the occurrence, lipid normalized concentrations. As Dr. Samsul already explained shortly, is chlorobenzene used as pesticide, herbicides, toilet deodorizers, industrial solvents, and many places, many uses and in industrial types. So the occurrence in fish lipids can be used as an indicator of the exposure, how much the fish were exposed to those chemicals. And for bioconcentration, we can continue to the toxicity. is that the levels of exposure enough to cause non-lethality or lethal effects, death to the aquatic biota. And then the third level, the third step is to calculate the hazard quotient HQ based on biota lipid. Risk assessment experts normally try to find the uncertainty, to decrease the uncertainty between those obtained in laboratory experiments. You can expose fish in like several days in laboratory conditions, and after that measure the concentrations in water over time, and also catch some fish and measure the concentration over time in fish lipids. And then we can make the ratio between the concentrations in the fish and concentrations in the water. You will get the KBL, bioconcentration factor. Chlorobenzenes are part of neutral lipophilic organics that mostly found in lipid tissues. But I think 20, 30 years ago, people more concentration or more focus on the concentration in water, lethal concentrations in the water, LC50. But these compounds is highly lipophilic. It means that their concentration in water are much lower than those in the fish, even up to 10 to the power of 6. One comparison 1 to a million. So using biota much more efficient than using the water to analyze concentrations in the water. Next slide. That's part of introduction. We have 12 congeners of chlorobenzene. This is a chlorinated compounds starting from 1 chlorine monochlorobenzenes with 3 congeners of dichlorobenzenes. And 1 for dichlorobenzenes, you may find it as toilet deodorizer, even in domestic exposure. So we have each 3 of 3 chloro and 3 tetrachlorobenzenes, and 1 pentachloro and hexachloro. Hexachlorobenzene, the most highly lipophilic chlorobenzenes, and this compound hexachlorobenzene or HCB is one of the PBT, persistent bioaccumulative and toxic compounds, and special concern in global level, including fish in aquatic waters, in aquatic environment. Next slide, please. And this is my latest research and publications. Normally, we compare the value not on a lipid base, based on body weight. This means concentrations of chlorobenzenes in per fish body weight, per gram of fish weight, or per gram of dry weight of fish weight. But now I try to use the concentrations well normalized to lipid content. So the title is Comparison of Lipid Normalized Bioconcentration Factors of Chlorobenzenes in Fish. Here some formula in the left side you may see. If you work in the lab, we get the KBL. It means concentration in fish lipid divided by concentrations in water. We're doing it in bioconcentration experiments in the lab. While we're going to monitor or survey in the field, we can get concentration in fish lipid and concentration in seawater or lake water. So you divide it, you get the CRL. Field to lab comparisons is always an important assessment in aquatic risk assessment. So if they are mimic each other, the uncertainty is very small, so the graphs would be very similar. As you see in the graph, log KOW as the axis, chlorobenzene starting from 3 to 5.5 for hexachlorobenzene. The higher the log KOW, the higher the tendency to bioaccumulate in aquatic biota, because KOW is measured as the concentration in octanol phase divided by concentration in the water phase. So you get 3 parts here. models, chemical models, fish experiment, fish biology experiment, toxicology in the lab, and in the field situation. If we can compare the three, the ideal models, chemical models, the fish lab experiments, and the field monitoring, so it will be give more and higher certainty to the risk assessors. You see in the graphs, there are three lines. KBL is a laboratory data of chlorobenzenes, quite close with the CRL, except in the higher chlorobenzenes with 4, 5, 6, they're getting close, while they're quite different with dichlorobenzenes, and until now, no data in monochlorobenzenes. And as you see in the right side, is compared to liposome water partition experiments, liposome water partition, and also triolein water. So we can see some comparisons between laboratory, field, and chemical models. Next slide, please. That's the exposure concentrations. And how about the lab? The toxicity data. The toxicity data, as I mentioned before, normally used as lethal concentration 50 measured in water. But now it was expressed as ILC, internal lethal concentrations. So it's more definite, more specific, not the concentration in the water, but concentration in internal tissues of fish. So because this concentration are highly lipophilic, it means the concentration in the water are very low. And even for hexachlorobenzenes, until now, we don't have toxic data of hexachlorobenzenes in water for fish. But we have the internal level concentration data with experiment lab data in the dot lines and in the lower lines, compared to the field data, ILC 50L. In environmental risk assessment, the difference by a factor of 1 or 1 log scales is acceptable while we compare the lab to the field situations. So we can see here in your right side and in the dark blue, that's the laboratory data, ILC 50L, internal, and compared with the models with the red color, that's the difference 10 to 11, 1 log scales. While the top two box, the SL solubility and the maximum concentrations in fish, that's the chemical models. That's the chemical scientists have provided the maximum solubility of those chlorobenzenes in the lipid. And then CBmax can be calculated using the log KOW. So KOW, octanol water partition coefficient, maybe if some of you not been accustomed to toxicology, this is the main parameters comparing the accumulation in octanol to water, simulate the accumulation in fish, aquatic biota, aquatic plants to the water. Next slide, please. And finally, we get the hazard quotients. Hazard quotient higher than 1, it means, or in the log scale, you see that's the lines posing significant risk and posing not risk. As we can see, 1 to 1,000 or 1 to 10,000 actually for hexachlorobenzene. That's how the managers of the environmental managers can use this data. How do they interpret this finding? This finding, HQBL or HQW, subscript W, hazard quotients in water, measured as concentrations of chlorobenzenes in water divided by LC50 in water. If the ideal situation should be HQBL is equal to HQW, but that's not in the case because HQ biota lipid is measured by dividing the concentration in fish lipid to the internal concentration in fish lipid, internal lethal concentrations. And then we can see that the ratio, both HQW and HQBL, much lower than 3 to 4 log scales. So the environmental managers can be confirmed that hexachlorobenzenes and in 1 to 10,000, I mean, to provide a risk is quite safe. Even safer for lower chlorobenzenes, especially for dichlorobenzenes, the range is 5 log scales, 10 to the power of -5. It means that, I mean, created less problems for dichlorobenzenes in the aquatic environment for current situation. That's the 3 summaries of my previous research. And then I have some framework of proposed idea. Let us look at in the next slide. Next slide, please, Regina. Yeah, this is normally biology and environmental scientists try to do. Try to external concentration in water or sediment, or can be adjusted to sediment lipid. And then we find in the red box in the middle, internal concentration in muscle, lipid adjusted. But in muscle lipid does not mean in the brain of the fish as the target site organs or in the livers of the fish. So environmental toxicologists catch the fish and measured in the muscles and adjusted the concentration in the muscle weight to the lipid muscle weight. But the main target may be not in the muscle, in the brain, in the livers, in the kidney of the fish. So we need response specificity. So that's why environmental toxicologist goes molecular. It means that try to describe the response in ecosystem level using molecular approach. So the response can be classified into these 3 categories: cellular molecular response in target site, organismal response, means like lethality, death of the fish, decreased number of population as an ecosystem response, or the decrease of bird weight, the decrease of liver weight. That's the type of organismal response. So here in these complex situations in the field, we need 3 questions to be answered: high concentration and response specificity in the molecular cellular level, which species is more sensitive in the organismal response, and of course, relevance in ecosystem situation, especially for Malaysia and Indonesia. We are islands, archipelagos, lots of fish and other aquatic animals. I can say that in the because we adjusted to lipid concentration, that's we can say in exposure lipidomics, and toxicologists also response in toxicolipidomics. So means ecosystem goes to molecular level. Next slide. That's the idea. We can see lipid both function as internal exposure medium as now also as toxicity target organ. The second one has not been much described in the literature. The lipid as internal exposure medium. In the old-fashioned or the traditional system, we use the concentrations in water, the external concentration in water to measure the safe to the fish, lethal concentrations in water. Now, it has been changed, or it has been converted, or we can say complemented. It has been complemented with ILC, internal lethal concentration, but in body weight. Furthermore, it has been developed into ILC subscript L lipid, internal lethal concentration in body lipid weight. And then possible in the near future, it will be transformed again to more specific ILC-TSL, internal lethal concentration in certain or specific target organ based on lipid, like brain lipid, kidney lipid, liver lipid, and so on. While in tissue histology departments, I mean, we can divide in the toxicologies between membrane lipid and storage lipid. Membrane lipid and storage lipid. They are two different roles in biochemistry, and especially for chlorobenzenes classified as non-specific toxicity. Some scientists propose the idea it influence the fluidity of membrane lipid or change the structure of membrane lipid, and then the fish has changed also suffer from change in lipid metabolism. And there is one paper divided, I mean, doing research comparing storage lipid and membrane lipid, as you may see in the next slide. Storage lipids water and storage lipid water and experiment and calculation level by Gessler. Nice, almost 1:1 the ratio data, and this can be used as a good starting point to further develop ILC-TSL. Lipid as toxicity target organ. Lipotoxicity has been defined as lipid intermediates can accumulate in non-adipose tissue, especially kidney and livers, and causes metabolic syndrome of change of lipid metabolism. Next slide, please. Next slide. Yeah. In ASEAN countries, Indonesia, Thailand, Malaysia, Philippines, there are many publications concerning Anguilla eels bicolor, especially Anguilla bicolor species. I searched and found that this type of eel has very high lipid content, more than 20%. Normally, in environmental toxicologists, lipid content of 5% will be enough. And that's the average lipid. We can say the low lipid content is less than 1% maybe. Classified as low lipid content, 1 to 2, less than 2%. 2 to 5 is moderate. More than 6, even up to 23.75% is pretty high. In European eels, even higher. It can be up to 40% of European eel consists of lipid. And then we have lipid composition by Ravendra. Dr. Ravendra has identified 25 fatty acids in types in lipids of Anguilla. And lipid distribution in organs of swamp eel. I think that's found in Malaysia, I think I forgot, or Thailand. Found that visceral organs as 4.56, skeletal muscles 1.8, and livers. And that sum up about 9% of swamp eels in Southeast Asia. And by Cyril Rakosopon, the quite difficult name. Yeah, here as we see in the ASEAN countries, ASEAN waters, we can see how these fish, these eels, distributed widely and found in all of our ASEAN countries, Malaysia, Brunei, Philippines, Indonesia, in many regions of Indonesia. And it's quite good bioindicator to be used in these ASEAN waters. And the most interesting part of Anguilla species we can use as their life cycle. They move from oceans to fresh water. So it means that they can be used as indicators in both marine waters and fresh waters. And already some idea or some research about the effects of climate change on Anguilla species. even north to Japan. There are some publication discusses the migrations of these eels from Southeast Asia to Japan in certain season. Next slide, please. Here in Southeast Asian regions, we found that some expert Chai and Arai found that the age from 5.5 to 10.5 years. It's quite long lifetime. It means they are good as bioindicators because their lifetime is quite long, 10 years, compared to other fish, which is less than a year, let's say. And at least there are eels already studied. Indonesia has lots of type of species. Philippines 5 and Malaysia 2, based on the reports by Chai and Arai. Interesting if we can know which one of those species of, actually there are 18 species in Southeast Asian waters. Yeah, I mean, even more in numbers compared to our in tropical Atlantic Ocean. We are in tropical Pacific Oceans. And here in Indonesia, we can see the distributions explained, reported by Sugeha, how these eels, 18 species, distribute in Indonesian waters. And eel ecology, starting with this, we can say the life cycles start from maturation and then further reproduction and temperature acclimation and even migratory patterns. In toxicology, it can be measured. What is the concentrations of chlorobenzenes or lipophilic organic compounds that relate to internal effective concentration, IEC, subscript L, of maturation? And we have not got the answer because it has not been studied yet. So it is a good direction to develop such kind of research. The same thing we can say, we can obtain the levels that influence the reproduction, acclimation to climate change, the rising of temperature, and the migratory patterns. As ecology, we can see, I mean, the movement of eel from one areas to another, the patterns of movement. But in lipidomics, in molecular science, which gene or which fatty acids more responsible during this migration season? And then in maturations, which lipids more responsible for juvenile to develop into mature and ready for reproduction? So ecosystem changes can explain better with such lipid analysis. Next slide, please. Here my summary. From Angela fish ecology and biochemistry, we get migration age 5 to 10 years, quite long. Maturation age, lipid content pretty high, body mass enough to be used as environmental samples. Usually we use 2 grams of dry weight. It means that around 10 grams of wet weight, but this one is adequate. And lipid distribution, we already get how they distribute in internal organs and muscles, and lipid composition. From my research, there's a small contribution into this big idea. Lab field model, the comparison within a factor less than 10, quite acceptable in ecological risk assessment, but we do need smaller numbers to reduce the uncertainty. And the membrane lipid and storage lipid, the toxicity of narcotic compounds as like chlorobenzene has been explained by Geisler and Esther. and increasing applications of lipidomics in environmental toxicology, especially in numbers of papers has been identified very significant increasing in the last 15 years. So we can get some ideas that Angela eels and other eel species are feasible for biomonitoring of persistent bioaccumulative toxicants, PBT, highly lipophilic and toxic compounds like as pentachloro and hexachlorobenzenes, highly relevant tissue samples, adequate amount of sample collection and analysis. So biology, biologists getting more higher important positions because we can describe the relationship between internal exposure in tissue lipid and internal responses, all in lipid-based measurements. And if that need further description of cause and effects, we can apply lipidomics and toxicolipidomics approach that can link cellular organismal population levels. And because of the area of habitat and migrations of those eels in ASEAN waters, so it will be fruitful for ASEAN countries to partnership together, to work together, collaboration, not only in environmental issue, environmental safety, but also in fishery, because it's one of the main food consumed by our people, and also for public health. Because some ASEAN people consume this as a main diet, and also especially for our friends in the North, Japanese, they like to eat eels. So many purpose, from molecular to ecosystems, and will give benefits not only to environment but fishery and public health. So that's the purpose of larger idea based on my research. Thank you, and I give back to the moderator. And happy to receive your questions.
Okay, thank you very much to Dr. Johan. Very interesting subject to discuss. So now we move on to the question and answer session. So I think we have a few questions here.
Yeah.
Okay, Dr. Johan, the first questions from Dr. Jacob. In the level of application, what types of fish can be used for biomonitoring or bioindicator of toxicity?
Yeah. Thank you, Mr. Jacob. We normally use, like catfish, fish in the bottom feeder, getting from sediment exposures. If we want to analyze more relationship with fish in the bottom parts of the lakes, water, or sea, we try to use bottom feeder fish. It means the partitioning of three phases: fish, water, and sediments. But if those fish live in water column, not quite close to the sediment, it represents the habitat in water and the fish bodies, the two. Yeah, these compounds, of course, deposit in the sediments as well, because this is the dumping site, or the repository, and can give high exposure. As a developing country, we need to be cost efficient. We should design monitoring standards. Let's say if those water bodies used for fishing, either by fishermen or tourists, it's quite important. We do some assessment using bottom feeder fish, because there's a prolonged exposure in the sediments, and we can use that type in the monitoring in the field. But of course, we should design to maximum concentration of certain compounds in fish. And interestingly, we should divide into two. For native people which diet almost every day, 3 times a day, they consume the fish. And for common people or for tourists, maybe once in a month they come and fishing, they get very few large exposure compared to the native people. So by designing such type of biomonitoring program, cost efficient, we can protect our people much better than we set a certain procedure. So we should know in which area in Indonesia, let's say, currently intensive fish lakes, intensively fish lakes. We should define first intensively fish lakes and rivers. And in those areas, we try to have 2 cat species, bottom feeder and column water fish. So we can get the whole ideas of the accumulation in the longer term, let's say 20 years from now. Thank you.
Right, thank you, Dr. Johan. So I hope that will explain. So we move to the second question. So question by Dr. Wan Rosiano. Dr. Johan, for information, Dr. Wan Rosiano, she's a biochemist, a colleague of mine here. So her question, may I know, do you also do toxicology study on any enzymes in fish? What are the species of fish that you usually study?
Yeah, I developed my research especially not for any specific type of fish. Even, my dissertation relate to partitions of chlorobenzenes in the sediment water system. I view all in interactions, sediment, water, and fish. And it is very hard to selectively, you should select which kind of fish species. Not like in the Northern America. They have like salmons and they have certain fish species to be used related to their daily diets. But in here, I think while I was doing some literature search, I found that because let's say we can start from biology perspective first or practical application first. Based on my analysis, Indonesia, very large country, need lots of fund budget to monitor its environment, both in freshwater and marine waters. And to monitor and to estimate the toxicity, we need certain amount of samples, and it depends on high lipid contents and highly lipophilic compounds. So how do we meet these two in the concepts first? In the fish, they should be high lipid content, and in the toxicants, they are highly lipophilic organic compounds. Okay, and they can meet each other here in this concept. And how to explain more? That's why my next step. Yeah, I haven't done in doing select certain fish or certain enzymes, but we should further investigate these tropical eels. What kind of specific enzyme responsible for migration, especially for maturation in tropical waters? Which enzymes and which genes responsible for maturation? And then after that for reproduction. And after that for migration pattern. And it is interesting if I can work together with friends from UITM in biochemistry and molecular science, that how we can say the molecular ecosystem in practice, from molecules to ecosystem, not only in the concepts, but in practice as well. Yeah, I like to do that, such approach. Thank you. But for now, I've done any experience in that specific enzyme.
All right, so thank you very much, Dr. Johan. So I hope Dr. Wanuziano can speak with Dr. Johan further about collaboration. Dr. Johan, I have a few questions. Okay, just a simple one. Do you think if you compare between the seawater versus the freshwater fish, so can we see? I mean the levels of differences between those two areas?
Yeah, a very interesting question, very important questions, because these chlorobenzene are lipophilic, not neutral lipophilic organic compounds. The influence won't be so much different in terms of chemical speciation or in terms of aquatic chemistry. It means we can use comparable marine waters and fresh waters. That's quite different if we say metal ions, and the metal speciation can depend on organic ligands, maybe more sulfur in certain areas of the coast, and they can influence the metal ions concentrations in those two areas, because metal ions that mainly partition or enter the fish tissue, but not for organic compounds. I mean, the difference may be quite not so much. It means they're very efficient. And keep in mind that catadromous fish means they migrate from ocean to the rivers. In Indonesia, some people have identified like 20 main rivers that those eels migrate. And we can check of the seasons, and we know the migration pattern followed by maturation level means they're ready to be consumed. And we can design sampling time. For public health protection is the time when they grow up and bigger size. And then we should search, especially while they do migrate they're growing up in fresh waters, and they should go back to the ocean for their reproduction, fertilize, for eggs delivery. So it means the higher size should be getting close between estuary or fresh water. And then the time while they grow up, you can get the idea. So it is certain interesting parts where the eggs level, juvenile or adult. Of course, for public health protection, we more care on adult levels. But for, I mean, our partners from Japan, they already like track the fish movement from Southeast Asia and back to Japan. Maybe they already more developed. They need those kind of analysis. Thank you. So interesting. Even Malaysia and and Indonesia separate by some marine waters in North Kalimantan. In your areas there are many rivers, and sounds good. And Brunei too. There are some good research published from Brunei University of Brunei in that area. So that's why. And in Philippines too. Philippines, more Japanese eel migrate to Northern Kalimantan and Philippines Islands. Yeah, that's it. Thank you, Dr. Samsul.
All right, Dr. Johan, one last simple question, because I didn't see any more question in the chat box. So I just have one last question. So what is your personal comment? People are now moving to use zebrafish to study toxicology.
Yeah.
So what is your personal comment then?
Yeah, based on your faculty direction of research, maybe we know SPMD, semi-permeable membrane device. Why don't we design and manufacture SPMD chemical lipids? To trace, to accumulate very small concentration of lipophilic compounds in the water bodies. So they move to the SPMD, but that is a non-living abiotic chemical approach. But why don't we manufacture SPMD made on fish or eels lipids, lipids of eels, natural lipids. So I can simulate, the lipids from the eels. That's the one. And the second two, because you're very advanced in molecular science, and maybe you may develop genetic markers, biomarkers, genetic markers for those ecotoxicity effects on lipid molecules. So I know from few of our technology department, we are dealing with more products as an expression of our theoretical of our sciences. So it's good. I mean, when I read lots of paper, SPMD and genetic markers from more developed countries, maybe that's more relevant and more affordable for us. If Indonesian government, let's say, want to use SPMD in remote areas, we can have in affordable costs made by our scientists. And how don't we try to develop SPMD or genetic markers, very rapid diagnostic tests, and can be used in our large marine and freshwater bodies. Thank you, Dr. Samsul.
Okay. So I think there is no more questions. So thank you very much, Dr. Johan, for your very, very interesting topics about aquatic toxicity. So I think I'm going to pass the session back to Dr. Shireen and Ms. Andrea. Thank you very much.
Thank you, Dr. Herman Johan, who explained very well the interesting material about aquatic reassessment, and also Dr. Samsul, who guide us as moderator.
Ladies and gentlemen, I hope you have found this webinar's presentation informative and inspiring. Right now, I would like to get everyone's attention and cooperation to switch on your respective cameras so that we can take a group photo of all speakers and participants. Okay. I might have to take a few sessions. So I will start with the first slide. Everyone's ready? 1, 2, 3. Okay, next. 1, 2, 3. Next slide. 1, 2, 3. One more. 1, 2. 1, 2, 3. One last one. 1, 2, 3. All right, thank you. Over to you, Andrea.
It is my pleasure to announce that we have reached the end of our collaborative webinar.
I hope you have enjoyed the program, and here's to more future collaborative programs between FSG UiTM and FSG UKSW. Thank you all.
Thank you all.
Thank you.
Thank you very much.
Thank you
very much,
everybody.
and all participants.
Thank you,
Thank you,
Professor
Farida. Thank you, Shirin.
Thank you, Ibu. Thank you. We did it. Thank you.
Ibu.
Thank you. Thank you.
Superb MC, superb moderators.
Thank you, Prof.
Thank you, Prof.
Well done, everyone.
Students.
To the participants, the link of attendance is in the chat room, so you can fill in for your certificate. Thank you.